Hello viewers, welcome to CEC Gurukul lectures. I am Dr. Sanawar Soham from Department of Botany, Kalindi College, University of Delhi, and I will be discussing significance of plant systematics and its relationship with other fields of biology to solve taxonomic problems. To begin with, let's discuss what are taxonomic evidences. In plant systematics, evidence is the information used in context for a purpose such as identification or classification. The foundation of plant taxonomy was laid on number of characters or attributes of plants and plant groups studied from time to time. Such taxonomic information can be realized in many ways to prove a hypothesis, solve a problem, characterize a taxon, classify a group of plants, or derive evolutionary relationship among plants. The gross morphology provides the foundation of plant taxonomy, and it is supplemented by the information from various other fields of botany. Now we'll move on to the understanding of alpha and omega taxonomy. The scope and methods of taxonomy have evolved with times. However, the principal core of the discipline has remained the same. That is the concepts, the classification of plant and plant groups, and nomenclature. To achieve these objectives, the approaches adopted are broadly classified into two kinds, that is alpha taxonomy and omega taxonomy. Taxonomists such as Valentine and Love in 1958 and Davis and Haywood in 1963 have suggested that the developments in taxonomy occurred in four phases. These are the first one, the pioneer or exploratory phase, the systematic or consolidation phase, the experimental or biosystematic phase, and the encyclopedic or holotaxonomic phase. According to this classification of developments in taxonomy, the first two phases, that is the pioneer phase and the consolidation phase, are more or less equivalent to alpha taxonomy, while the phases 3 and 4, that is the biosystematic phase and the encyclopedic or holotaxonomic phases, are similar to omega taxonomy. At the level of alpha taxonomy, the organisms are characterized, identified, classified, and named. The pioneer or exploratory phase of taxonomy is basically analytical. It involves collection of plants for analysis of their characters. Therefore, in this sense, it is a pioneering effort for understanding the plants and hence the name. At the same time, it is also considered as an exploration of nature. Hence, this is regarded as the pioneer or exploratory phase in taxonomy. This relates to basic or preliminary classification based almost entirely on external morphology. This can be considered as the empirical approach in taxonomy, where the classification is synthesized from the observed facts. The vast amount of data collected by the taxonomist must be evaluated and then consolidated. This leads to the development of systems of classification. Thus, the pioneer or the exploratory phase progresses to a systematic or consolidation phase. Now, alpha taxonomy has also been called classical or orthodox or formal taxonomy. This has been practiced since ancient times, and different aspects of this approach are in use even today. After synthesizing a basic classification, the taxonomist then attempt to improve upon it. Therefore, the observed facts are interpreted in omega taxonomy to provide an interpretive classification. Also, the evolutionary and phylogenetic approaches are applied to understand the taxonomic and evolutionary relationship of plants at all levels. This can be achieved either by experimentation or use of large number of features such as cytology and genetics for evaluating taxonomic relationships. Thus, developments in taxonomy involving detailed cytological and genetical studies become important. Now, this has been referred to as experimental or biosystematic phase. It was then realized that knowing all aspects of biology and chemistry of plants would provide definitely a much better understanding of relationships. Thus, collecting and collating information from many sources as available led to the recognition of the encyclopedic or holotaxonomic phase in taxonomy. Omega taxonomy has also been called as beta taxonomy, neo taxonomy, or modern taxonomy. So, the taxonomic evidences are the characters or attributes which are gathered from variety of disciplines. Most of these characters are used in describing patterns of variations at or below the species level. The sources of such characters could be from morphology, anatomy, embryology, palynology, cytology, biochemistry, cementades, molecular biology, etc. Now, I will move on to the taxonomic evidences from palynology, but before that, we shall understand what is palynology. It is the study of pollen and spores. The taxonomic characters of pollen grains include the wall structure, the polarity, symmetry, shape, and grain size. In angiosperms, pollen grains are of two kinds, that is, monocolpate and tricolpate. In this slide, you can see that monocolpate pollen grains are boat-shaped. They have a long germinal furrow and one germinal aperture. This is characteristic of primitive dicotyledons such as Piperaceae and Chloranthaceae, and also majority of monocotyledons, cycads, and seed ferns. According to some palynologists, the first flowering plants had monocolpate type of pollen grains. Now, talking of another type, that is tricolpate pollen grains, as you can see in the slide, they are globose, symmetrical, have 3 germinal apertures, and are characteristics of advanced dicotyledons. Some of the characters of the pollen grains that constitute taxonomic evidences are its unit, polarity, shape, symmetry, nuclear state, wall architecture, exine stratification, exine structure, and sculpture. It also includes shape, structure, position, number, and type of apertures. All these features really help to solve many taxonomic problems. This slide shows the pollen grains from various culturing from various plants. Now, if we take up some examples that how palynology has helped as a taxonomic evidence, the first one being the pollen grains of Magnolidae. They are binucleate, whereas those of Caryophyllinidae and are trinucleate. In Ericaceae, the pollen grains occur in tetrads, while polinium are characteristics of Asclepiadaceae, Apocynaceae, and Orchidaceae. Now, if we talk of another example at the family level, within the family Betulaceae, the exine of pollen grain in section is knob-like in Betula, whereas club-shaped in Corylus, unexpanded in Carpinus, arch-like in Alnus. Within the family, Epicardiaceae. This is another example where the pollen grains of the genus Brachyloma are solitary, whereas those of the genus Epactris are in tetrads. Moving on to the next example, where pollen apertures have helped to solve the taxonomic puzzles in different species of the same genus. For example, Bauhinia acuminata, it has pillate type of pollen apertures, whereas striate in Bauhinia kurgai and echinolate in Bauhinia malabarica. The pollen grain size could also be used to identify the species of a genus. For example, the pollen grain size in Malva rotundifolia is 74 to 84 micrometers, while it is in range of 105 to 126 micrometers in another species of it, which is Malva sylvestris. An interesting example of palynological evidence in taxonomy is of genus Trisygen. This plant has separate male and female plants, which is known as dioecious state. On the basis of morphological evidences, the genus was placed in the family Euphorbiaceae. However, when female plant was discovered, it was transferred to family Fagaceae. Later, when the pollen grains of male specimens were examined, they were found to be exactly similar to the member of Fagaceae. So now, this genus Trisygen is regarded as a member of the family Fagaceae. Some taxonomies still suggest the name Nothophagus for this genus and classify it in the family Nothophagaceae. Now, we will move on to another example where embryology is the study. I will also discuss now few examples how embryology has helped in solving the taxonomic problems and acted as taxonomic evidence. But before that, we shall understand what embryology is. So, embryology is the study of successive stages of sporogenesis, gametogenesis, and the growth and development of embryo, endosperm, and seed coats. Although the possibility of utilizing embryological characters in taxonomy was indicated by some earlier botanists like Hofmeister and Strasburger, the role of embryology in solving taxonomic problems was first brought in prominence by a German embryologist, Schnarf, in 1931. Now, I will discuss some examples from embryology. Now, embryological evidences have been used in solving taxonomical problems almost at all levels and have helped resolve the doubtful systemic positions of several taxa. However, according to Jones and Latzinger, the embryological characters have proved to be of significant help in determining relationships within the families, genera, and species, but they are less useful at the rank of, say, order, subclass, or class. The determination or delimitation of angiosperms in the first instance has been largely based on embryological characters, we all very well know. They are characterized by enclosed ovules that are mostly anatropous, polygonal type of embryo sac in majority of flowering plants, exhibits double fertilization and triple fusion, and post-fertilization development of polyploid endosperm, which also supports the probable monophyletic origin of angiosperms. Now, the further primary classification of subdivision angiospermy into two classes, namely monocotyledony and dicotyledony, is largely based on the characteristics of embryo, which are almost constant for the group. Now we will take up the examples where embryology has helped in solving the problems above the family level. there are few orders of angiosperms, the members of which are morphologically well defined and also exhibit a set of uniform embryological features. For example, caryophyllales. Now, it is more widely known as centospermae and is embryologically very distinct. The order is characterized by trinucleate pollen, bitegmic crassinucellate ovules, which are campylotropous or amphitropous, commonly with the inner integument longer than the outer, and very often with a space between the integuments towards the chalazal end. The seed has a curved peripheral embryo, more or less surrounding the food storage tissues, which consist mainly of perisperm with little or no endosperm. Moving on to the next order, Lacithidales, this order, which was formerly considered to be related to myrtales, has been found to differ from the latter by the presence of alternate leaves, centrifugal stamens, lack of internal phloem, and a series of embryological features in its various members. Then another example is of Ericaeals. It is a homogeneous group of sympatalous families that includes members, most of which share several common embryological features, including notably the production of pollen in tetrads. So, this feature very well helps in solving and finding out which order it pertains to. Moving on to Orchideales, this order is characterized by undifferentiated embryo and very little or no endosperm. Now, we'll take up some examples at and below the family level. Now, if we talk of family Onagraceae, it is characterized by the presence of peculiar monosporic 4-nucleate onagrate type of embryo sac in all its genera except Trappa. Such an embryo sac is derived from the micropylar megaspore of the tetrad. The lower 3 chalazal megaspores do not disintegrate. In this type of embryo sac, antipodals are absent. Different systematists have placed the genus Trappa in several ways. Some have placed it in Onagraceae, some have placed it as an isolated member of Holaragenaceae, and some have placed it as only genus of monotypic family Trappaceae. Embryological evidences, however, strongly support the last view that it should be placed in Trappaceae. Trappa has a well-defined suspensor hostorium in addition to the 8 nucleate embryo sac, which are absent in any other members of Onagraceae. So, we can very well find out Trappa and we can very well identify Trappa with all these features, embryological features. Also in Trappa, the ovary is semi-inferior, bilocular with one ovule in each locule, and the fruit is a large one-seeded drupe, while in other members of Onagraceae, the ovary is inferior and tetralocular with numerous ovules in exile presentation, and the fruit is generally a locidutical capsule. Now, moving on to the next family, Podostomaceae. It is a remarkable family and exhibits a variety of interesting features. Members of this family are recognized because of the formation of pseudoembryo sac, which is formed by nucellar cells. So, this helps in finding or identifying Podostomaceae from rest of the families. Specific examples of the role of embryological data, if we talk of Paeonia, many taxonomists have treated this genus as a member of monogeneric tribe Paeonia of the family Ranunculaceae. But Paeonia differs from Ranunculaceae not only in its chromosome number, vascular anatomy, floral anatomy, but also in embryological details. such as Paeonia shows a unique embryogeny with its young zygote first becoming cenocytic, and then the cell formation begins from its periphery. Now the embryo initials develop from some of the peripheral cells, and the cenocytic phase, although common in the gymnosperms, is unknown elsewhere within the angiosperms. The seed of Paeonia are arylate and fruit a follicle, while in Ranunculaceae the seeds are non-arylate and the fruit is an achene. So the embryological details thus suggest that Paeonia belongs to an independent family, Paeoniaceae, instead of Ranunculaceae. Now let's move on to another branch, which is cytology, that has helped in solving the taxonomic problems and acted well as a taxonomic evidence. Now cytology is the study of morphology and physiology of the cells, and the information about the chromosome number, shape and pairing at meiosis is used for the classification purpose. Cytotaxonomy refers to the use of chromosome number, morphology, ploidy level, ploidy type, chromosome aberrations as data for classification. And cytogenetics includes those studies dealing with the observation of chromosomal pairing or behavior at meiosis. So let's move on to the importance of chromosome number, structure and behavior at meiosis as the taxonomic evidence. If I talk of chromosome numbers, we are generally aware that the number of chromosomes in each cell of all individuals of a single species is constant. It is also established that the more closely related species are likely to have similar chromosome numbers, while the more distantly related ones shall have different numbers. Due to this relative conservativeness, chromosome number becomes an important and frequently used taxonomic character. In addition, I will give you some example. There is a wide range of chromosome numbers in the angiosperms, from as low as 2n, that is the diploid or somatic chromosome number equivalent to 4 in Haplopappus gracilis, which belongs to family Asteraceae, to as high as 2n equivalent to 530 in Poa literosa, which belongs to family Poaceae. A large number of angiosperms have been analyzed for their chromosome numbers, providing useful taxonomic information. Now, in the later lecture, I will be discussing few examples of the chromosome numbers that have helped to solve the taxonomic problems. Thank you.